right now? वीडियो ऑन करूया. Hello, good morning, everybody. In this rainy morning, we welcome you all in the Rajaram College, Kolhapur for this national seminar on research panorama of life sciences. Today we have with us the honorable guest and speaker of this today's session, Dr. Prashant Kodgire sir, IIT Indore, our honorable principal, Dr. A.S. Khemnar sir. I welcome you all on the behalf of Rajaram College, Kolhapur. Now I will hand over to Dr. J. A. Savan ma'am for giving introduction of our today's guest.
Hello. A very good morning to all respected teachers and my dear students. With a great pride and honor, I am here to introduce our today's speaker of National Seminar on Research Panorama of Life Sciences, Dr. Prashant Kodgire sir, IIT Indore. Presently, he has working as Associate Professor, Ramanujan Fellow, Center for Biosciences and Biomedical Engineering, IIT Indore, MP. Sir has completed his master degree in microbiology from Government Institute of Science, Babasaheb Ambedkar, Marathwada University, Aurangabad. He has received his PhD in biotechnology from IIT Bombay. Sir has also postdoctoral fellow University of Chicago in the year 2008 to 2012, Department of Molecular Genetics and Cell Biology, Chicago, USA. Dr. Kodgire sir worked as a research associate, Oakheart Research Center, Biotechnology Group, Aurangabad. He has also received many grants from different funding agencies for many projects. Dr. Prashant Kodgire sir received many awards and honors. There are so many awards and honors. The first is the Ramanujan Fellowship from Department of Science and Technology, Government of India. In Irvington Institute, Postdoctoral Fellowship from the Center Research Institute, USA. International Postdoctoral Fellowship Award from the Lady Tata Memorial Trust, UK, for research in leukemia. Senior Research Fellowship from the Council of Scientific and Industrial Research, CSIR. Junior Research Fellowship from CSIR. Eklavya Scholarship from the Government of Maharashtra, India, for excellency in academics. I end up with this brief introduction of Dr. Prashant Kodgire sir. Now I request to sir start the session with his lecture, and the topic is Molecules of Life and Central Dogma of Molecular Biology. Thank you, sir.
Am I audible?
Yes, sir, you're audible.
Great. Can you see my slides?
Yes, sir, we can see your slides.
Okay. Thanks to organizers for having this webinar session. I understand that all of us are working from home. Few of us are going to office, but in this negativity of COVID pandemic, staying connected to science, learning new things, probably gives a lot of positivity. And that is very, very important, that along with keeping our health, our mental peace is also equally important. So I really congratulate the organizers for having this webinar series. And it's indeed a great privilege to be there with other eminent panelists like most of you were there for yesterday's talk, and I am big fan of Professor Rakesh Mishra, who is director of CCMB, and you got some glimpses of what is happening in the area of genomics. Today, we will go a step backward, and before talking about genomics, we'll talk about the thing which you already know, which you probably have studied. That is, what are the different molecules of life and the central dogma of molecular biology. So as it was introduced that I am a faculty member at IIT Indore, Madhya Pradesh. And this is a second generation IIT started in 2009. And our department was started in 2012. Before we were discussing about actual presentation, I asked the organizer what's going to be the composition of the audience. And I'm told that there are lots of biologists and there is a significant number of non-biologists also. So there are some research scholars, PhD students, and there are teachers. So because this audience is going to be a really, really diverse audience, I will try to keep my lecture as non-technical as possible. And obviously, for few people like hardcore biologists, this is a refreshment and a revision for what they already know. And for those who are not familiar with modern biology, is some new learning for them. So we'll have, let's say, like 45 minutes of discussion, and then we can have some questions. Now, when we say molecular biology, this is a bit recent term, but term biology is not new to us. Most of us are aware that human beings are fascinated to understand the biological processes. So when we say molecular biology, all of us are interested to understand what is happening with our body, what is happening with our environment, what is happening with our surrounding. So the air we breathe in, the food we ate in the breakfast, or how different functions work, how heart is pumping the blood. You are currently listening to it, how this listening mechanism is happening. You are also watching the slides in front of your device. So what kind of signals are generated, how your eyes are capturing it, how these signals are sent to brain. So there are so many different things which are happening in a biological body, biological entity, and we are interested to know mechanisms behind these different functions. So molecular biology is nothing but understanding biological functions at a molecular level. And this is not new to us. Our ancestors were also interested to understand what is that which is important, what is that is controlling us. And then this is a classical textbook picture which is showing structural organization of a cell. This complex functions which we do are also carried out by a complex structure which is there inside a cell. All of you are familiar with what I am trying to show you here. So there are lots and lots of compartments which are dedicated to a particular function. Okay, so the way our house is organized, that we have a kitchen, a living room, a bedroom, and a bathroom, and a visitor's room. Similarly, you have a complex architecture inside a cell where there is a nucleus, there is endoplasmic reticulum, there is a Golgi apparatus, there are mitochondria, which are energy creating organelles. And the current understanding, because you have studied that the genetic material, the master molecule, is sitting inside a cell. But this understanding what we have right now was not there, let's say 2-3 centuries ago. And people were proposing different theories that what could be the exact mechanism for how life is generated. Okay, what is life? How life is generated? How cell is divided, or how an organism is divided? These were all mysterious questions for people, and therefore people were interested to ask questions. So, what is the uh, genetic principle that accounts for transmission of traits from parents to offspring? Human life has a long history, and different civilizations, and they were trying to ask these similar questions, that what are the genetic principles that account for transmission of traits from parents to offspring. So in short, they were trying to ask what is that which is called as heredity, which is responsible for that, why kids or son or a daughter is looking like their parents, why there is matching of skin color or the height or the physical appearance and morphology. That's not true for human beings, that's true for animals, that's also true for plants. And among different theories which were proposed at that time, one possible explanation they come up with, that hereditary is something called as a blending hypothesis. So they were not aware about the genetic material which is regulating and deciding the morphological features which they could see by their naked eyes. But they think that there is something, some material which is controlling life from parents. It is getting mixed in a manner analogous to the way you mix two different colors. All of you know that when we teach our school kids, that how you make a green color, you mix a blue color and a yellow color, and then you generate a green color. So that is what was the blending hypothesis. But there was an alternative to this blending model that was called as particulate hypothesis of inheritance, where they propose idea of a gene. Okay, so the parents, they pass discrete heritable units or genes to the subsequent generation. That was the idea. And all of us know that the fundamental work was carried out by this particular gentleman, Roger Mendel, and there are lots of misconception about the work carried out by Mendel. People know that he was an Austro-Hungarian monk, and he did experiments on pea plants, who happened to become foundation of modern genetics. And therefore, we call Mendel as a father of genetics. But let's go and see what was the background behind Mendel's work. Why a monk, so-called monk, started doing experiments on pea plants. So that's only part of the story. If you read the contemporary history at the time of Mendel, what was happening, the geopolitics happening at that time, you'll understand the real reason for Mendel's work. So this was the time of colonization. Most of the countries in Europe, they learn how to do navigation. They started building ships, and then this is like in 16th century, 17th century, they started going out of their area, that is Eastern or Western Europe. Now, all of you know that Eastern and Western Europe is typically a cold climate. You have some diversity there, but once they start learning navigation and they started going towards other continents, such as Africa, Asia, and they also started, they were interested to come to India. India was connected, Indian merchants were going there and they were selling our products over there. But Europeans started going to different parts of world only in that particular phase which is like 16th and 17th century. Now, when these people started going there, they saw different culture, they saw different plant varieties, they saw different animals, and they realized that those plants and animals, they have distinct property. Some of them, they are high yielding varieties of cow or sheep or goat, and there are crops which are high yielding crops with different taste, different flavors and different texture. And then they were, which were not aware about these different characteristics, they were interested to know and study it. So they brought and collected different seeds from different parts of countries wherever they went, and invented new varieties. And they also started study. So there was a competition among different countries of Europe, not the present Europe. The countries were there, but they were like mixed boundaries. So there was Spanish people, there were Portuguese, there were German, there were Dutch. All these people, they were interested to study what is happening with these particular traits. So this particular Austro-Hungarian Empire, they were also not far behind. And these people, they formulated a society of bright young people in that particular country. And they were told that please come join hands and let's start doing some research on understanding the different varieties. Most of them, they started doing the so-called hybridization or breeding experiments. And then they realized that when you mix two things, something else is coming out. And that is what also inspired Mendel to do what he did. So he studied Pisum sativum in the beginning of 1854. And he painstakingly worked for 11 years. Okay, so understand that how much patience one has to have to do some fundamental work. So he worked for 11 years. And then 1865, he published his work as Experiments in Plant Hybridization. So this work was published, but society at that time was not ready to accept what Mendel is trying to propose. Please remember that Europe was dominated by so-called Christian theology. And the Christian theology was telling that no, world is created by God in 7 days. And they also tell you that on which day what was created. Now, when there is a theory of that, okay, everything is controlled by God, the church was dominating the mindset of people at that time. And then Mendel is going against the flow and telling that, no, that's not true. And he started giving his laws of traits and segregation, which all of you are aware about. But this background about Mendel, you are probably, everybody is not aware about. So as I'm trying to tell you that he worked for 11 years, published his work in 1865, but society was not ready to accept what Mendel was trying to tell. Okay, so his work was not at all accepted and recognized by scientific community and public at large at that particular time. But what he was trying to tell was the truth. And you cannot hide truth for a long time. So Mendel's work was rediscovered in early 19th century. Okay, so ideas of inheritance by Mendel's times were vague. So people were not really following what he was trying to tell, because Mendel was ahead of the time. He was quite ahead of what was the thought process of that particular time. And therefore, his work has to wait for almost 35-40 years for people to recognize it. And I'll not go into these details which you have studied into your class book. This is there into school textbook also, that how he worked on 7 different traits. He was lucky that he worked on those traits which happened to be there on different chromosomes. all of us should also appreciate that the way Mendel designed experiments were too tight. He has done lot and lot of homework, and these were the positive experimental results which were published. But at the same time, he must have done a lot of other experiments which probably helped. So those people who are happen to be a PhD student, research scholar, understand that how you have to have lots of patience. You have to work hard to do some fundamental discoveries, the way Mendel worked for 11 years. Then only you can do something or checking you can do, which is going to change the worldview. I'll not go into details about what was happening, but all of you know that only one thing, that he did some experiment with purple and white flowers, the yellow and green sheets or round or wrinkle sheets. And by and large, he found that in the F1 generation, the ratio of dominant versus recessive character was 3:1. Those who are biology students, they know it very well. Those who are non-biology students, they have studied this when they were in the higher secondary. These were the Mendel's conclusions. What he was trying to tell, genes are the distinct entities that remain unchanged during the crosses. The hybridization or cross experiments he did, he's trying to tell that genes are the distinct entities that remain unchanged during the process. One more big thing he was trying to tell, which now we appreciate, that our genome is not a haploid genome, but it's a diploid genome. Mendel in his work in 18th century told that each plant has two alleles for a gene. So in short, he was trying to tell that our genome is a diploid genome. When he was not aware about what is genome and what is the chemical nature of that, but he told that there are two alleles. This was very, very significant. This was very, very different. And then further, he told that alleles are segregated into gametes in equal proportion, and each gamete got only one allele. Now, you know what we are talking about, that meiosis, where a haploid genome is making a diploid genome is making a haploid gametes. This is what Mendel proposed long time before. And then what he told is, during gamete fusion, the number of alleles are restored back to 2. Remember, Mendel never had sophisticated instruments the way we have today. He was just basically doing theory. He was only able to do experiments based on what he could see by naked eyes. And the marvelous thing what he could do, even we are not able to do same thing with the modern technology and instruments we have with us. How beautifully he told and did those painstakingly experiments. Imagine that those who are following botany, they know that doing these cross is not easy. You have to remove stigma or stamens and then collect pollens from one plant with a paintbrush and then do pollination, cross pollination. You have to cover these bags, flowers with bags, so that once you do that experiment, no cross pollination should happen, because insects will come and you screw your experiments. So he was aware that what he was doing. He did wonderful design of his experiments, and what he proposed, that yes, we have a diploid genome. In gametogenesis, we make haploid gametes, and in fertilization, gametes fuse to restore the two genes or two alleles back. This was something which was really, really wonderful. However, people were not aware about what is the nature of genetic material. They were able to see things which are phenotypic, which are morphological, which can be seen by naked eyes. All of you are aware about this puppet show, that you see puppets are dancing, but they are not dancing on their own. Somebody is controlling and choreographing and deciding that at what particular time, what is going to be movement of that particular puppet. So we see that so many different functions. we reproduce, we eat, we see, we observe, we feel. So our sense organs are doing so many different things. But how these things are controlled and what is the nature of genetic material was a big question for biologists in, let's say, 19th century. Mendel told that there is something which is controlling, but the chemical nature of these controlling element was not aware. And there were again two different theories. Chemistry was a bit popular, and when people started studying a cell, they realized that when you work with a cell, you have different components, such as you have nucleic acid, you have proteins, you have lipids. So there were two different groups. One very popular and dominant group was in favor that the exact nature of genetic material must be protein. Because when they studied chemistry of these molecules from a cell, they realized that, oh, it's just a nucleic acid. It looks very similar. There are no modifications. There are no differences. And then we see proteins where there are hundreds and hundreds of different protein molecules. So it is very logical for them to connect that, oh, there are different functions. There are different functions means there are hundreds of functions and there are hundreds of protein. So if there are lots of function, lots of protein must be required to control these functions. So many favored that protein is the main genetic material which is controlling the cell. Because DNA is simply 4 nucleotides. So how could it hold such a complex genetic information to control that complexity, what we are able to see? Contrarily, proteins are much more complicated. Chemically, they are made up of 20 amino acids. They are more abundant, and perhaps they hold the genetic information. And there were no conclusive evidence whether so-called genetic material or gene or is protein or it is DNA or nucleic acid. And all of us, we know that these were some seminal work done by Frederick Griffith. All of you are aware about it. And subsequently, the work carried out by Avery, MacLeod, and McCarthy that conclusively told that the genetic material is nothing but the DNA and it's not protein. For those who are non-biologists, I'll just explain you very briefly that I told you that in 1865, Mendel proposed his law of genetics or inheritance. But in 1902, his laws were rediscovered, his work was recognized. From 1902 to 1928, people were not aware about what is the exact chemical nature, and there was debate going on. And it is in 1928, Frederick Griffith, he did experiments with a bacteria known as Streptococcus pneumoniae. Now, Streptococcus pneumoniae is a causative agent for pneumonia, and there was some work going on that. It has two different strains. One was called as smooth strain, which happens to be a virulent strain. And there is another strain which will not cause a disease. So therefore, it is called as non-virulent strain. So he did some bacterial transformation experiments. And with those experiments, they realized that it is indeed that DNA is the genetic material. So what was his experiment? I told you that this Streptococcus pneumoniae has two different forms. So you first injected this smooth virulent strain of bacteria into mice. And as expected, the mice was dead. He was also aware that if you do culturing of this smooth and a potent or a virulent strain of bacteria and grow it for multiple times, you are able to convert it into a rough or a non-virulent strain. And because it is non-virulent, you inject in the mice, mice is alive, nothing is happening to it. And the beauty of his experiment comes here. So he had a smooth cell, he heat killed it. Because you are killing that bacteria, you inject it alone, mice is alive. Okay, this is very straightforward. You inject a live virulent bacteria, mice is dead. You inject a non-virulent bacteria, mice is dead. You kill the smooth bacteria or a virulent bacteria, and therefore mice is still alive. But if you mix this live non-virulent bacteria with heat-killed cell suspension of the smooth bacteria, the virulent bacteria, the combination of these two is injected in mice. And there is a surprise that this mice is dead. And when he did the autopsy of this dead organism, he was able to recover the smooth bacteria. Remember that he never used live smooth bacteria. He started with rough bacteria, but he was able to get the smooth bacteria. And then he realized that there is something into the dead cell components of the smooth bacteria, which is transforming characteristics of this rough and making it smooth. And that was nothing but DNA, which was subsequently done by these marvelous experiments by Avery, MacLeod, and McCarthy, where they treated these heat-killed samples with either protease, which will kill protein, which will destroy protein, or an enzyme called as RNA, which will destroy RNA molecules only, or DNase, yet another enzyme, which will destroy only DNA, or they were able to separate only by fat by ultracentrifugation. And then these separately, individually treated rough cell lysate was introduced, smooth cell lysate was introduced into rough cells. And they realized that when you destroy DNA, R cells remain R, and R cells are not transformed into S cells. And this what was confirming that it is indeed the DNA which is transforming rough cells, non-virulent cells into a virulent strain. This was a big discovery for biological world, that we are able to find out the master molecule, the one which is controlling a cell, one which is deciding how the cellular functions are regulated. The next big milestone which came was in 1953. I told you that 28, Frederick Griffith did some experiment. 44, we were aware that it is the DNA which is master molecule, but the exact structure of DNA was not known to us till this seminal paper in Nature, which was published in April 25, 1953 by two stalwarts in this area. All of you know, Watson and Crick, where they told that the exact molecular structure of nucleic acid. Remember that in 40s and early 50s, lots of big scientific groups, very popular scientific group, there was a mad race to find out structure of DNA. There are two popular groups which were working, but all of us know that it was Watson and Crick who proposed the DNA as a double helical structure. That was in 1951. James Watson learns about the X-ray diffraction pattern projected by DNA. And then they were also aware about the chemical composition of nucleic acid, that it is composed of a sugar, that's deoxyribose sugar. There is also a phosphate, and there is a nitrogenous base. They were also aware about that the ratio of A and T is equal to ratio of G and C's. So why always A is equal to T or G is equal to C? People were not able to answer that. But when they proposed this model of anti-parallel double helical structure of DNA, then they were able to solve this mystery. Why A is always equal to T and G is equal to C. So this was this figures from their paper. They created a DNA fiber. It was extracted by X-ray beam, and they were capturing the image. This was the real image which is published from their paper. Okay, this was showing that there is a diffraction pattern. And just by looking at this X, they were able to propose that DNA is a double helical structure, and it is an anti-parallel double helical structure. Same time, Linus Pauling and Robert Corey at Caltech, they were also in a race to propose DNA structure. They were also close to tell something, but they were not able to tell what Watson and Crick were able to prove. This is a picture when both Watson and Crick were young, and these are their more recent pictures. And I feel privileged and very happy that when I was at University of Chicago, I work in the same building where James Watson also worked. So, and believe me, I could feel vibration of those great personalities who were there. So as I told you that this was that particular picture, they diffracted the DNA fragment, and then they realized that it is that DNA is double helix anti-parallel. All of you studied this in your textbook picture, that this is a ribbon model and this is the space field model, a ball model. There are two anti-parallel strands which are made up of sugar and a phosphate. They go into opposite direction, and these two strands are connected by nitrogenous bases: adenine, thymine, guanine, and cytosine. Two of them are the purine, two of them the pyrimidine. Purines are double ring structures, pyrimidines are single ring structures. So always a two ring structure is joining with a single ring structures. These two strands, they wrap around each other every 30 angstrom, once every 10 base pairs. And as I told, that they are held together by hydrogen bonds between adenine and thymine. There are two hydrogen bonds, and guanine and cytosine, there are three hydrogen bonds. A few years back, to honor Rosalind Franklin, the Rosalind Franklin was the one who developed the X-ray structures for DNA. And to be precise, the photo number 51 from her work was used by Watson and Crick to crack the secrets of DNA as a double helical structures. Now, this is a slide for non-biologists, which tells you about how a nucleotide will look like. There is a nucleoside where there is a sugar and a nitrogenous base. And when you add phosphate to it, you can have a long chain which is connected by sugar and phosphate. So again, nucleic acids, there are two kinds. There's DNA and RNA. Deoxyribose sugar is there in DNA, and ribose sugar is there into RNA. The fundamental difference is there at carbon 2 position. This is H group, and therefore it is called as deoxyribose. This is OH group, that's why it's called ribose. And these are the nitrogenous bases which are there into DNA and RNA. Purine are double ring structure, as I mentioned a while ago. The two of them, adenine and guanine. Pyrimidine are single ring structure. Cytosine and thymine is there into DNA, whereas cytosine and uracil is there into RNA. Let's come to the main topic of today's talk, that is the central dogma of molecular biology. After Griffith in 30s told that DNA is genetic material, and in 1953 when Watson and Crick proposed model of double helical structure of model of DNA, then people started asking question. If protein are the major working force on the ground and DNA is genetic material, how the information which is present onto DNA is coded onto protein? It's a big mystery. Also, if DNA is genetic material which is master molecule, which is controlling all the cellular functions, then when cell is dividing, how cell is controlling and how it is making another copy of DNA. So all of we have studied this in our books, that DNA is copied back into another copy. That's process of replication. This DNA is transcribed into an RNA molecule or messenger RNA called as transcription. And this RNA is translated to form a protein. And there is a lot of coordination between how this DNA is transcribed into RNA, RNA is translated into protein, and how these proteins themselves are required for process of replication. They're required for process of transcription, and they also play a very important role into translation. So very briefly, we will discuss and refresh our memory what is happening. Now, there are two distinct cells we have studied in our biology textbook. This is a simple mechanism, simple depiction of a cell called as a primitive cell or a prokaryotic cell. Now, what is this cell? This is a bag of membrane, cell membrane, with some nucleic acid and some proteins. Okay, there's a bag filled of different cellular component, different similar cellular chemicals. So there is a DNA, which is a chromosome or genetic material. It is transcribed into a RNA, and this RNA is translated by a machine called as ribosome to synthesize protein. This protein will have different functions. They can act as enzyme. They can have different structural roles to give integrity to this particular. But the more evolved organisms are called as eukaryotic cells, where there are compartments. Remember that very first slide which I showed you, where there are complex organization, where I was able to show you nucleus, the Golgi apparatus, endoplasmic reticulum, there are mitochondria. All those components and compartments make this particular cell quite complex. Here, the genetic material, the master molecule, is located into a distinct component called as nucleus. And therefore, replication is happening into nucleus and transcription is also happening into nucleus. mRNA is made. This mRNA, it comes out into cytoplasm, where yet another complex and beautiful machinery, ribosome, will translate this RNA into protein. Let's go and find out what was happening. Another beautiful experiment which was done by some stalwarts in this area, that indeed replication is a semi-conservative model of replication. All of you know Messerson-Stars experiment. Biology students, you can wake them up in the midnight and they are able to tell this particular experiment how it was done. For non-biologists, I can explain you in a minute that very simple chemistry was used. You use radio-labeled media components, the raw materials used for biosynthesis of protein and DNA, and then you ask some questions that how DNA is copied, how DNA is made into another copy. So when people were not aware about the exact mechanism of replication, there were multiple theories. There were basically three different theories. People were talking about that the original copy of DNA is not unchanged. It is remain untouched. An exact same copy is made without touching the first copy. And that model was called as conservative model. And there was a dispersed model. People were also aware about that genome is big, such as human genome, whose size is in billions of base pairs. They were also curious that how you can have such a big genome copied without any difficulty. And therefore, one more model was proposed that this particular DNA is made into pieces. These pieces are copied and they, by some magic, it is assembled back. And there was yet another model that was telling that it is a semi-conservative model. The two strands of DNA, they separate by some mechanism. And each of these two strands, they act as a template to synthesize a new strand. So among these three different models, which one was correct was not sure. And as I told, there was this semi-conservative model where this pink color parental strand is separated and blue color strands are made. Or it is the conservative model where this pink color parental DNA is kept untouched. And without touching it, you copy that information to make the new blue. Or it's a dispersive model where you somehow break into pieces and then you assemble it back. And during assembly, you can have a mixture of pink and blue together. So as I told you that Messerson-Stark, this experiment, they did this experiment with N14 containing media. all of you are aware that I told you that there are nitrogenous bases into nucleic acid. These nitrogenous bases are necessary components of DNA. Without that, you cannot have DNA. But you grow this particular E. coli cell into a media where you don't provide regular nitrogen, but it's a radioactive nitrogen. And because it's an N14 nitrogen, if you do centrifugation, it is heavy nitrogen. Sorry, N14 is the regular nitrogen, which is a cold nitrogen. But if you grow these cells into a radiolabeled nitrogen, N15 nitrogen, you get sedimentation at a lower level. So you grow these E. coli cells for lots of time so that all its DNA is labeled with N15, N15, and therefore in ultracentrifugation, it will show band at a lower level. You passage these E. coli cells, which are grown on N15, and transfer it to N14 containing media. You allow it to grow for only one generation, and then do the same experiment. You isolate genomic DNA and find out what is the sedimentation coefficient of this particular DNA. And believe me, this particular DNA, which was coming at bottom of the tube, started migrating upwards. They did this experiment for another 30 minutes, and then they realized that this band which migrated, it started showing you two bands. And all of you are aware that this is how the classically they could see it, that you were having first the heavier band, which was shown here in N15 containing media. You did transfer, and after one particular replication cycle, you had a hybrid DNA, hybrid of N15 and N14. And then one more cycle was giving you two bands, a hybrid band and a light DNA band. And after many different growths, you will see a very light band of a hybrid DNA and dominantly a light DNA. And this was that experiment by Mrs. Stark. Just by doing simple radioactivity, differentiating between DNA, by doing density gradient centrifugation in 6 molar cesium chloride, you are able to differentiate their sedimentation coefficient for N15 DNA, N14 DNA, and a hybrid DNA of N14 and N15. This was a very fundamental discovery. You know that DNA is double helical, you know that these are anti-parallel, but how it is copied, how parent is making two copies for making two daughter cells. Messerson star DNA was the important step for all of us. So all of we know that the replication is performed in a very meticulous fashion. There are 3 steps. There is a initiation, elongation, and termination. And this process is highly regulated at the origin by some regulatory protein. This decision to go for replication or not is tightly regulated. Cell will not take a random decision. And the main enzyme in replication is DNA polymerase, which will add a new nucleotide based on the guidance from the parental strand, the denatured template strand. I'll not go into details because biology students are aware about it, and non-biology student, if I try to tell you and explain into minute detail, we need a lot of time. But it's a complex mechanism where you are initiating it by initiator proteins, and some enzymes like primase will come, make a RNA template, RNA primer, and then on top of that, you have a replication bubble and you make a copy of DNA. I'll switch gear and talk about a distinct phenomena which happens only into eukaryotes. It doesn't happen into prokaryotes. Prokaryotes, you are making a RNA, and that RNA is immediately going for translation. But I'll talk about something which happens into a little bit evolved organism, that happens into eukaryotes. If DNA is copied into another DNA, is a straightforward thing. If DNA is copied into RNA, that is also not very difficult. is the same principle where DNA template is used as a guide and a RNA molecule where the only difference is that thymine is replaced by uracil and sugar is not deoxy, it's a ribose sugar. There are no interruptions. Gene is copied into mRNA and mRNA is translated into a protein. But that's not true for people like us, human beings, which are little bit evolved. Our DNA is not directly matching with the protein because it is interrupted by something called as introns. So the protein coding regions, which are shown here by colored boxes, they match with the mRNA, but there is difference between mRNA and protein. So what I'm trying to tell you here is that the human genome, which you probably got some glimpses in yesterday's talk by Professor Rakesh Mishra, is a complex entity. And that particular gene, the basic fundamental unit of a cell, is interrupted by some portion of DNA, which are not going to code for any protein, So this particular DNA, when it is transcribed into RNA, it also has those exons which are going to go and code for protein, but it also has those interfering regions or called as introns. And there is another beautiful mechanism by which these introns are removed, and that is called as splicing. Let's spend a couple of slides on this process of splicing, and non-biologists will be very happy to understand how such a complex process is controlled in our cell, which is happening right now. When we are attending this webinar, there is replication which is happening in your body, there is transcription which is happening in body, and almost every mRNA, pre-mRNA is spliced out. Now, people have tried to find out what is the length of those intervening regions, what is the size of exons, and most of the biology students, they are probably aware about it, that the average size of exon in human is around 200 base pair, but for worms and fly, it is a bit longer. Okay, so we say that fruit fly, Drosophila or C. elegans, they have longer exons, but little bit evolved like humans, they have shorter exons. They also tried to find out what is the intron length, and you will also imagine that intron length is also varying between humans, C. elegans, and Drosophila melanogaster. So what do you see here? Humans, they have by and large intron length of around 100 to 2 kb. See, more than 50% of introns of this length. But some of these interfering DNA fragments are more than 30 kb. So one exon is sitting to another exon by a distance of 30 kb. So it's a large piece of DNA. So how cell knows that what is the boundaries of these introns and exons? How cellular machinery recognizes what part to be cut and joined back? And another big scientist and big discovery laid by a scientist called as Philip Sharp, who published this PNS paper in 1977, told us that what is happening with a process called as splicing. Remember, this paper is published almost like 40 years ago. So biology is one such science where it is evolving very fast. Every year, you have some old hypotheses which are demolished and new theories are proposed by people. So until this paper by Philip Sharp, people were not aware about what was exactly happening in splicing. So he did some experiments with this adenovirus, and this was the actual electron micrograph he published in his paper. And based on this figure, he made this cartoon depiction and told that the genomic DNA and mRNA, they are not matching exactly, but they are matching into patches. And which are those patches? The patches which are exonic, they are matching, they are retained into RNA, mRNA, but there are intervening regions which are shown in the yellow color. They are the introns, they are not there into mRNA. And because of his seminal work, Philip Sharp was awarded Nobel Prize in 1993. I'll not go into more details of it, but I'll just briefly tell you that subsequent work by many, many people, we were able to tell that there are distinct definition of where exon will end and where intron will start. And similarly, where intron will end and new exon will start. Okay, so all these introns will always start with GU and they'll always end with AG. They are there in all our pre-mRNA, they are there in all our genes. There is hardly any gene in humans which don't have an intron in it. We'll not go into philosophical discussions about what is requirement, why we have introns, whether it is wastage of energy, why we are copying all this information many, many times. That could be a topic for another seminar, but people are aware about the splice junctions, where exactly is exon ending, where exactly intron is starting, and there is a complex mechanism where these introns are removed by something called as transesterification reactions, where you remove this lariat kind of a structures and two different exons represented by a red box and a pink box are joined together. Okay, this is a very beautiful mechanism. Now, people, just to summarize that, this process is called as splicing, is performed by the most complex macromolecular structure known to human being. People were talking about the replication machinery or transcription machinery is complex. Then people came to know about the ribosomes are complex, but spliceosome is much more complex than ribosomes. They're made up of snRNA and many other proteins. How many? There are more than 300. Okay, and as I mentioned, that this makes the most complicated macromolecular machine known to us till date. So again, we'll not go into more details about why spliceosomes are extremely complicated, why there are so many different proteins, like 300 different subunits. But I just want to tell you that exact reason why complexity is there is not known, but understand that there are at least 4 different reasons why you have a spliceosome such a complex structure. One is that the defective RNAs can cause a lot of problems to cells. So there could be some subunits from these 300 which might assure that the correct splicing will happen and there are no errors retained. One more thing is that you don't want to transfer an unprocessed RNA outside for translation. It will translate to a random protein. And splicing is tightly coupled to the nuclear transport or nuclear export of that processed RNA. So you require some accessory protein which will make sure that only right processed mRNA will go out. Splicing is also coupled with transcription. So you have to have some crosstalk between the transcription machinery and splicing machinery. And some proteins may act as messenger between these two different processes. And a very important reason why you have such a complex machinery is that many of these genes, they are splice alternative function. Okay, so one gene is able to generate many different proteins. And I'll give you some such examples how that particular happens. All of you have studied that one gene will always give one protein, but splicing is telling you something different. Splicing is telling you that one gene can give you several proteins. See how beautifully you are able to generate variation just by having one gene is able to give you multiple different. Now there are other reasons for why you can have one gene and multiple protein. One is that you can have an additional or an alternate polyA site. You can have an internal promoter which is responsible for giving you a different mRNA. But the main reason for getting multiple different protein is something called as alternate splicing. There is yet another reason for getting multiple protein from one gene is what is called as RNA editing. So if you want to summarize what is alternate splicing, you have these exons which are arranged, and there is a different possibility that you remove these introns which are by a very tightly regulated process called as spliceosome. But while removing, intentionally, you will skip some exons or you will retain some introns, and you can have different permutation combinations. Additionally, you can have multiple splice junctions. There are weak splicing junctions, there are strong splicing junctions, and cell will choose whether to use 3' splice junctions or 5' splice junctions, and combination of all of these, intron retention, exon skipping, alternate 5' exon selection, and multiple promoters and multiple polyacid, you can have variations of mRNA from same gene. And here is one such example where you can very well understand what is happening. Now, this particular example is of a gene called as CAMKD2 gene, which is encoding for an enzyme kinase. Now, there are 17 different exons, and these exons are alternatively spliced. And therefore, this particular protein, this particular kinase can be directed at different cellular components. How is that achieved? Imagine that this exon number 13 is skipping exon number 14 and directly going to 15, 16, and 17. This particular exon 15 and 16 is having a signal which is targeting this particular kinase to the membrane, because it has that particular signal as a transmembrane domain which is coded by exon 15 and 16. So this particular mRNA, where there's a delta A particular RNA, is targeted to mRNA. On contrary, you just retain exon number 14, don't bring 15, 16, and directly go to 17. Now this exon 14 happens to have yet another signal, yet another barcode, which is a nuclear localization signal. And therefore, this kinase is now transported to the nucleus. And if I don't have signal or barcode for membrane localization or nuclear localization, I do alternate splicing where I bring exon 13 and directly join to 17. No signal, it will remain into cytoplasm. So just by selecting and picking right signal or right exon, one particular gene is making 3 different proteins. One can be targeted to membrane, another targeted to nucleus, and yet another which will remain into cytoplasm. This is done by alternate splicing. And here is yet another example, a protein called as troponin T. Now this troponin T, there are 18 exons. And luckily, exon number 4, 5, 6, 7, and 8, they are alternatively spliced. And therefore, all of you study basics of mathematics. By permutation combinations, you are able to generate 64 different combinations. 64 different combinations of troponin T genes. Now, all of these, they are expressed into a unique tissue. Some of them, they are expressed only, let's say, into muscle. Others are expressed into intestine or kidneys or different cellular organelles. But because of alternative splicing, from one gene, you are able to get 64 different variations and 64 different protein components. So, what is happening here is that there is variation which is created by yet another process called as RNA editing. I will not go into much more details, but I'll just tell you that there are few genes where the DNA and protein are not absolutely matching at all, because more than 50% of bases are edited, modified by process of RNA editing. Uh, uh, because of lack of time, I'll not go into detail, but they also have some role into it. So by just the process of RNA editing, you can control whether particular protein, ApoB, has 100 amino acid protein into liver, but the same gene, because of RNA editing, is going to create a stop codon, and therefore ApoB48 is expressed into intestine. Okay, this is what is because of yet another post-transcriptional RNA regulation that is called as RNA editing. And finally, in last few slides, I'll tell you about that all these processed RNA are translated, and all of us know that there is something called as genetic code. These are three base letter code which are used by yet another complex machinery called as ribosome, which is composed of two subunits. The mRNA in prokaryotes, it is there into cytoplasm itself. So transcription is making RNA, and RNA is immediately translated. But in eukaryotes, you have a nucleus where the replication and transcription is happening. And after transcription of that pre-mRNA, there are modifications happening such as capping, splicing, and polyadenylation. And there is a nuclear transport of that RNA to the cytoplasm, where this particular machinery, ribosome, will capture it and then translate into protein. So these are classical textbook images. What is the composition of bacterial ribosome? For non-biologists, understand that how complex they are. There are two different RNA, 23S, 5S, and there are 31 proteins into large subunit. And in small subunit, there is 16S RNA and 21 proteins. Mammalian ribosomes are more complex. There are more proteins and more RNAs into it. And they are responsible for generating the proteins, which are the main molecules for doing the actual function. These are the soldiers on the ground. They are the ones which are making as the backbones of different structures. So the big challenge for biologists was to understand that how the message which is present on RNA, a nucleic acid, is transferred on to yet another chemical moiety, that is protein. Remember that nucleic acid building blocks are nucleotides and protein building blocks are amino acids. Chemically, they are very different. So copying RNA to DNA or DNA to RNA is very simple chemistry because they are similarly made. But nucleotides and proteins are chemically very different. How can you have signals on nucleotide and decode them into making a polyamino acid peptide? Was a big mystery, and when people studied ribosome, they were able to know that the major decoding work is done by this beautiful small molecule called as tRNA or transfer RNA, where they are able to read this triplet codon and able to guide an appropriate amino acid for those codon. This particular figure will tell you that how there are 64 different possible combinations. 3 of them, they work as a stop codon. 1 will act as a start signal, and rest of them, they are going to code for 20 different amino acids. Why we are studying all this? Why we should know? This is a particular slide which will probably help you in understanding that this is circuit of our life. Biologists are telling about a circuit. What is the circuit? All these different dots are important metabolites in our body. And all these lines are the enzymatic reaction which are performed by at least one enzyme. If you misplace with this particular network, if there is a short circuit, there is a problem. All of you know that we have lots of electronics in our life, and because of one small mistake in that particular circuit, you have to change that particular chip. Same chip is there in our every living cell. These enzymes, which are coded, which are nothing but proteins, they are precisely regulated. They are supposed to perform the right function. This is the glycolysis, this is the Krebs cycle. If you change these enzymes, their function, their kinetics, it will change the whole metabolism. And therefore, you may have a pathological condition which will lead to a disease. We also know that apart from this metabolomics, we have other important cellular functions, such as the way our immune system works, which will allow us to fight against different infections. The ongoing corona pandemic, how the antibody diversity is generated, how we are able to recognize there is an infection, how our immune response is activated to fight against the invading pathogens. All those things are nothing but regulation of gene, and all these genes could be a yet another talk for us, where the genes are regulated at transcription level, at splicing level, at polyadenylation level, transport to the cytoplasm. There are regulation by small RNA. There are regulation which are there at the half-life of mRNA, how they are regulated at the translational level. The proteins which are made, they have their own half-life. So there is regulation which will decide how much gene is expressed, when that particular gene is expressed, and where that gene is expressed. So the quantity, how much, location, where exactly it is expressed, and when the timing of those gene expressed is a topic of yet another seminar. I will thank all the audience for your patient listening, and we can have the next course of action based on whatever the organizers have decided. Thank you very much for joining.
Thank you so much, sir. Hello?
Yes, please tell me.
Thank you so much, sir. So we will have there are some questions. I will be reading for you so that you can answer. Can I start, sir?
Sure.
Yeah. So the first question is, what is the function of intron in human DNAs?
I briefly mentioned about the possible reasons why we have introns. The exact reason people don't know why we have introns, but there are theories. Many people have many different theories, and one such theory is that which we didn't cover here, but all of us know that our genomic DNA or genetic material is prone for mutation. If we are prone to mutation, we know that change in DNA will change a nucleotide. Change in nucleotide of such diseases, such as, let's say, sickle cell anemia. All of you are aware about, even if you don't know minute details of it, but this is a disease, a genetic disease, where only one amino acid is changed because of one mutation in one particular nucleotide. So to protect DNA, it is fragmented into pieces. It is fragmented into pieces, and then you are able to protect those small pieces of DNA, or called as exons. That is one theory. Another theory is that I told you that you are able to create diversity. How do you create diversity? By a process called as alternate splicing. Troponin T example was one such example, that one gene is able to give you 64 different possibilities of protein, which are there into different tissue. Now that is possible because of alternate splicing. And alternate splicing is only possible if you have introns in it. So we have a gene, one particular gene is active, or isoform of a gene is controlled or expressed into one particular tissue type, but that is not there into other tissue type. So the alternate splicing mechanism by the spliceosome is able to generate that diversity of different proteins, because there are introns present, and therefore you are able to do alternate splicing. If you don't have introns, you are not able to do alternate splicing.
Yeah, thank you so much, sir. So the next question is, how helicase know the exact position at which it has to join and proceed to unwind the double-stranded DNA?
Can you repeat the question?
Yes, sir. How helicase know the exact position at which it has to join and proceed to unwind the double-stranded DNA?
Okay, so again, now we need a lot of time to describe about exact process and control at DNA replication level. DNA replication, as I mentioned, is divided into initiation, elongation, and termination. There are different challenges at every level, but the classical example is, let's say, a bacterial genome is initiated. A bacterial genome are initiated at something called as ORI, or origin of replication. Now, this is a unique architecture. There are unique sequences, there are unique repeats, and there is a particular chronology of event. Okay, so a bacteria, we feel that bacteria is so tiny and it don't know anything, but every bacteria, before it commits for cell division, assesses so many different things. It makes sure that there are enough nutrients. It makes sure that the surrounding environment is feasible for cell division. It also makes sure that there is enough energy and raw material for making a copy of DNA. So there is a complex signaling pathway which is integrating information from different sources, and that integrated information is communicated to few proteins which play an important role into DNA replication, which happens at origin of replication. So these proteins will go and bind to these specific regions onto DNA called as ORI, and they'll start unwinding it. So activation of these proteins, targeting of these proteins is a highly regulated mechanism, which is studied right now by many people, but that is what is precisely happening. So helicase is recruited after these initiator proteins are activated over there. And once helicase comes, it does. But how those initiator proteins are activated, how they are judging these signals of the energy level, the raw material level, the building blocks are there or not, whether cell is at the right cell division cycle, all that is decided very precisely, meticulously, and that decision is made because once you enter into elongation phase, there is no going back. There is no going back, and therefore most of the regulation for replication is at the initiation stage.
Yeah, thank you so much, sir. So the next question is, it is possible to produce any hybrid by joining human and plant DNA?
It's quite possible. It is doing what is what we call as genetic engineering. All of you are aware about Bt cotton. Now, what is this Bt? This is a protein called as cryprotein from Bacillus thuringiensis. That particular gene is taken from a bacteria and put into cotton plant so that that particular protein is expressed into plant. The pest will eat that particular protein and it will die. So it's a pesticide. Same way, we have human gene, insulin gene, it is expressed into E. coli. So the beauty about genetic material is all the living organisms, they have same chemical nature of genome. The DNA, if you isolate from different organisms, chemically they are not different, and therefore it is very easy for genetic engineers to copy DNA from one species and put it into other species. Most of the modern biology, we do it routinely into our lab. So it's very possible. We can have yet another lecture for how the fundamentals of genetics engineering are how we use different enzymes such as restriction endonuclease to take out a particular DNA, how we use polymerase chain reactions to amplify that DNA, how we use yet another class of enzymes such as ligase to put it back. So it's part of detail, but to answer it shortly, we can copy paste DNA from one organism and paste it into other organism very easily.
Thank you so much, sir. The next question it is, how one can use alternative splicing to cure disease?
Alternative splicing are happening in our cell and they're tightly regulated. However, the regulation, if it is not controlled properly, that will lead to expression of an unwanted protein at a higher level, or you have a protein which was supposed to be present into intestine is now made into liver or vice versa. Those things are already aware, and therefore people are trying to identify different targets and different novel ligands to control alternative splicing mechanisms. As I told, that splicing itself was discovered in 77. Alternative splicing was not like more than a couple of decades known. Many labs are trying to understand the mechanisms of alternate splicing, and they have come to a conclusion that yes, these splicing regulators have pathological role. There are hundreds of papers where people are trying to identify whether these splicing factors or splicing regulators can be used for drug targets. So it is possible. We can have different case studies. I cannot tell you right now a precise example until and unless I tell you background, what are those genes, what is their exact role, and how they can be used for controlling a disease.
Okay, thank you so much, sir. So there is one more question. Why and how tRNA can solubilize?
No, I didn't get this question.
Why and
tRNA
how can solubilize?
No, I don't think the question is framed properly.
Okay, so we will go for the.
Yeah, we can ask that person to reframe that question.
Okay, sir.
I am not
So
getting what exactly you mean by solubilization.
Okay, so we will go for the next question. Are ribozymes same as spliceosomes?
No, spliceosome is something different, where there are snRNA and other proteins. Ribozymes, one beautiful example is our ribosomes. Now, I told you that composition of ribosome is RNA and protein, but the exact the peptide bond formation, which happens between two amino acids, is performed by RNA. So ribozymes are the RNA molecule which will act as enzymes. And beautiful example is ribosomes. The transpeptidase reaction is not performed by any protein, but it is performed by RNA. And what is happening into splicing? Yes, there are transesterification reactions, and there are like 6 different RNA molecules working into it, but there is some role of proteins also there.
Thank you so much, sir. So the last question is, what triggers the process of splicing and formation of a certain mRNA from the hnRNA?
hnRNA are members and components of ribosomes. They are coded by independent genes. So hnRNA are not making mRNA, but hnRNA help into processing of mRNA. Now, how splicing is triggered? Every gene, as I told, is intervening of exons and introns, and you have to remove those introns, otherwise the frame of that particular mRNA will be distorted. If you because ribosome don't know where to start and where to what is the boundary of introns and exons. They only know that AUG is the start codon. They'll start adding amino acids and they'll end at the stop codon. So if you give a pre-mRNA, non-processed mRNA, you'll make a different protein. And that will have altogether different frame and therefore different composition. And therefore every protein has, every pre-mRNA has to be spliced. And therefore, when transcription initiation happens at the promoter, RNA polymerase is assembled, and the C-terminal domain of RNA polymerase happen to recruit components of spliceosome machinery. So until and unless spliceosome machinery is coupled to the transcription machinery, polymerization of transcription will not happen. The mRNA which will come out from the RNA polymerase is immediately offered to the post-transcriptional modification machinery. Immediately the capping enzymes will come and start protecting that mRNA. And the spliceosome complex will start identifying the intron-exon junctions. So the process of splicing and transcription, they happen in sync. They are happening together. And therefore, we don't need to say that how splicing will be triggered. They are happening simultaneously. As and when the RNA is synthesized, it is coming out of RNA polymerase. Immediately, the splicing factors and the splicing proteins and the respective RNA molecules will recognize and initiate the splicing process.
Yeah, thank you so much, sir. If you permit, there are two more questions that we will be taking.
It's up to you. I'm happy to take questions.
No, sir. There are only last two questions. Can we deal?
Yeah, please.
Do prokaryotes undergo alternate splicing?
Prokaryotes don't have introns in it. Therefore, there are no splicing happening. And if there is no splicing, there is no alternate splicing. Alternate splicing is there only into eukaryotes.
Yeah, thank you so much, sir. This is one more interesting question. Is it possible to make autotrophic human being?
What do you mean by autotrophic human being?
Yeah, it's like asked like that only. Is it possible to make autotrophic human being?
No, I am not able to understand what is autotrophic human being. So please request the person who is asking question to elaborate a bit more.
Just like I think it's they are going to ask just like plants, can there be autotrophic human beings?
Means what? What do you want humans to do like plants?
Photosynthesis to conduct photosynthesis.
Oh, okay. You want to have humans who can do photosynthesis, so we don't need to eat. We can just go and sit in sun and then get our energy. Again, like the way I told you that there is a metabolic circuit, the human circuit, that is there unique into plants and unique into humans. Theoretically, if you have all the enzymes required for photosynthesis clone into a human being, yes, but that's not that easy. Okay, we are finding it challenging to make a vaccine for coronavirus since last 4 months. Understand that. And even if we make that vaccine, it will take at least a year from now. So these experiments are not easy. They are painstaking. Theoretically, yes, you can have a genetically modified organism the way we have Bt cotton. The cry gene, which is not expressed in cotton, is now able to express. We have edible vaccine where banana is cloned with a particular vaccine and you just eat banana and you are getting vaccinated. You are asking cow to have a milk with spider protein in it. So all these things are possible, and similarly, you can have the whole photosynthetic pathway genes can be cloned into human, but it's quite challenging. When it will happen, I don't know.
Yeah, thank you so much, sir.
thanks for inviting me, and thank you for your patient hearing.
Yeah, I will like to hand over the session to Dr. S.V. Toro ma'am, the organizing secretary of this national seminar, to give vote of thanks of this session.
Hello. Namaskar. Respected Principal Dr. Khemnar sir, today's guest Dr. Prashant Kodgiri sir, participated teachers and students. I am thankful to Kodgiri sir who handled a highly interesting but simultaneously somewhat difficult topic for today's session, molecules of life and central dogma of biological molecules. He touched the topic by including different aspects of life sciences like organization of cell, organization of biomolecules within the cell, environmental interactions, and heredity. He started his main topic with Mendelian work, and with the help of interesting slides, he discussed the nature of genetic material. Sir has mentioned historical development in this field. He explained the molecular structure of DNA in more detail, along with the background of the workers in this field. He also shared his feelings regarding the workers because he worked at the same institute. He explained the relationships between DNA, protein, DNA, RNA in a very illustrative manner. He also mentioned modern techniques in this field like splicing, DNA-RNA hybridization, RNA editing, and protein synthesizing machinery, including ribosome. So he explained the session in very illustrative fashion. Today's session will definitely help to understand the metabolic network of the cell, changes in it, regularities developed by it, as well as irregulations in the metabolic network, which will hamper immunity and leads to disorders, which is very important in this today's period. The lecture will be helpful not only for biological teachers, but for all participants which are from non-biological subjects to understand the importance of science dogma and biological molecules. I am thankful to our enthusiastic principal, Dr. Khemnar sir, all my colleague teachers, participated teachers, and students. Once again, thank you, sir.
Thank you.
Thank you so much, sir. It's a very kind request to all the participants that the feedback forms will be given. The link for the feedback forms will be given in the last session, that is the 4th day of this national seminar. Thank you for your patience listening. Stay home, stay safe, and stay tuned with us for the next 2 days. Thank you. Thank you so much.